Hello, this is the Pinvision, and today we are going to talk about Mendelian inheritance and the pedigrees. When we discuss inheritance, we refer to the passing of traits from one generation to the next. The first man to attempt to describe such inheritance was Gregor Mendel, a priest who conducted experiments on the pea plants. He investigated 7 traits, for example, flower color, seed shape, seed color, and etc. During the experiments, he cross-pollinated 2 true-breeding pea plants with colors purple and white. We can call this generation parental generation. And to define the true-breeding specimens, they are the specimens which has ability to pass down specific phenotypic traits to the offspring. So, if two purple true-breeding specimens gets cross-pollinated, they will only give us the purple ones, and if white ones get crossed, they will give us the white ones. Now, after these 2 flowers were cross-pollinated, Mendel only got the purple flowers in the first generation, and as we can see, white color got almost lost. But was it really lost? So he decided to cross-pollinate the members of the first generation, and he got the purple and the white flowers with 3-to-1 ratio in the second generation. Now a question persists. Where did white color go and how did it appear? To explain the results, we need to understand that for white color to emerge in the second generation, there must be information about white color still contained within the generation 1, and since purple color was visible in the first generation and most of the second generation, we can assume that it is a dominant trait, so it dominates over the white one, and the white trait is the recessive. Now, to go into a little more details, we know that these kind of traits are written into the hereditary materials called DNA, which is organized into the chromosomes. Now, this DNA contains segments called genes. A gene is a hereditary unit that encodes RNA or proteins to create traits, and the allele is the different version of the gene. For example, let's discuss the allele number 1 of the purple flower. Now, as you can see, it has some kind of the DNA sequence which encodes to the protein which actually creates the purple pigments, and that's why the color is purple. But for the second allele, it gives us a defective protein which gives us the no pigments, which in result gives us the white color. So the different alleles constitute to the different traits. Now continue on. How did you get half of the information from the paternal side and other half from the maternal side? Because as you can see, for white color to pass on to the next generations, we have to get them from somewhere. Now, each of the flower creates a gamete. Now, if we look deep into the gamete, as you can see, it is made out from the 3 chromosomes, but only one of its kind. So there are no homologous or similar chromosomes, as you can see, and this is called a haploid karyotype, where n is equal to 3. So it is amount of the different types of chromosomes we have in cell. Now, when such a 2 gametes merge, they create a zygote. And if we look at the karyotype of the zygote, as you can see, we have the 2 homologous, and as you can say, similar, same chromosomes, one from the paternal side and one from the maternal side. And this is called the diploid karyotype. As such, we can define the first law of the Mendel, the law of segregation, which tells us that every individual possesses 2 alleles and only one allele is passed on to the offspring. Now, let's look more into the details of the notations, and if we choose the 2 homologous chromosomes and we choose a little segment, we can call these genes some kind of the Latin letter. This is called the gene notation. And for the dominant allele we use the big A, and for recessive allele we get the small a. Now we can use a Punnett square to actually define the possible zygote variations from given gametes. To quickly define also the other terminology, the bigger, so the homozygous individual is the individual which has the similar same alleles for the both chromosomes, and the heterozygous individual is the one which has the different alleles for the two chromosomes. Now, on the Punnett square, at the outside of the square, this is where we write the possible gamete genotypes of the parents, and inside the squares, that's where we write the possible genotypes of the offspring. Now, can you attempt to write Mendel's experiments using Punnett squares? And of course you can, and we will attempt it right now. So, for example, this was the first crossing, and this was the second crossing. And as you know, since they are the true breeding species, they should be both homozygous, and the purple one will be the bigger AA, and the white one will be the smaller AA. And as you can see, to write the first crossing, as you can see, we only get the heterozygous individuals with letters big A, small a, capital A, small a, and all of them will be purple. That's why we only got the only purple color, because the bigger A dominates over the smaller A, because as you can see, since we have the one gene which encodes the functional protein, we can still generate the purple pigments. Now for the second crossing, we merge the two homozygous individuals, and as you can see, we get 3:1 phenotype ratio, which was observed by the Mendel himself. And we also get 1 to 2:1 genotype ratio too. Now Mendel was curious about his experiments and asked, what if we observe two traits similarly, so how do they pass on to the next generation? For example, let's choose two peoplets. R will be the gene for shape and Y will be gene for the color, and let's choose two the heterozygous individuals with genotypes capital Y, small y with capital R, small r, and let's decide we cross them. What result do we get? So if they are completely linked, so they pass on at the same time, this is the possible gametes we have, because they are maybe on the same chromosome, or they are linked in other ways. And as you can see, this is the only possible variations, and this is only possible offspring variation. And as you can see, we get 3:1 phenotype ratio and 1 to 2:1 genotype ratio. But now let's discuss what happens if they pass on independently. Then we have the 16 different offspring genotype variants and 4 independent gamete variations, because as you can see, it's not necessary for the dominant alleles to be linked together, and in each gamete there might be a dominant allele with a recessive allele. This will be the possible offspring genotypes, and this will be the possible phenotypic visualization. As you can see, the phenotypic ratio will be 9:3 to 3:1, and genotype ratio would be this large ratio, as you can see. And when Mendel performed the experiments, he got the independent genes case. As such, you can define the second law, the law of the independent assortment. The alleles for different genes segregate independently of each other, leading to new combinations of the traits in offspring. Of course, this is the only case for the Mendelian inheritance, and as you will see in the future videos, the genes can be actually linked, and this was actually visible into the Morgan's experiments, and we will discuss it later on. Now, let's talk about the pedigrees. Now, pedigrees are used to visualize the inheritance patterns between the parents and their offsprings, and to define some roles. For example, white square is the unaffected male, the white circle is the unaffected female, the black circle is affected female, and the black square is affected female. And when using the pedigrees, the problems about the pedigrees will be mostly about guessing the genotypes of the affected people. Now, this is the end of the video. In conclusion, the Mendelian inheritance tells us about how genes pass on to the offspring. Law of the segregation tells us that we get different alleles from both of our parents. Law of independent assortment tells us that different genes pass independently of each other. Punnett square is used to find expected ratio of genotypes between the offsprings, and pedigrees are used to find the relationship between offsprings and parents. Thanks for watching, and problems will be down below the description about the stuff I explained. There will be upcoming video for solutions for that problems, and stay tuned.